Ethiopian Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on the microscope. We studied the microscope in our last lesson. We looked at the different types of microscopes, focusing on the light microscope and the electron microscope. We learned about the functions of both, and we discussed how they show specimens. Today, we will continue our lesson on the microscope. We will distinguish between magnification and resolution of a microscope. Let us begin the lesson with a review question. Students, please explain how the light microscope and the electron microscope work to form an image. You may discuss your answers with a partner.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, students. I am sure that was a simple activity for you. A light microscope uses a beam of light to form the image of the specimen. An electron microscope uses a beam of electrons to form the image. As we learned in our last lesson, microscopes magnify very small specimens, such as cells or tissues. The term magnification means increasing the size of an object. By observing the smallest forms of matter, scientists can learn as much as they can about organisms. Light microscopes can magnify a specimen up to 2,000 times, while an electron microscope can magnify a specimen up to 2 million times. Light microscopes are of great importance to scientists by enabling them to observe the living cells. But electron microscopes have enabled scientists to learn much more information about cells, such as their particular functions. This is because the electron microscopes focus on a magnified image, which permits the most detail to be observed. When an image is magnified, often it can be blurry or distorted. The term resolution means the ability to distinguish between two separate points. In other words, resolution means the amount of detail that can be seen. There is a minimum distance between two objects that is necessary for them to be seen clearly. If they are situated too close together, they often look like they are one object instead of two. The resolving power of a microscope determines how much detail the observer can see. This means that the greater the resolving power of the microscope, the more detail that can be seen. Light microscopes have a lower resolving power compared to electron microscopes. The magnification from a light microscope is limited by the resolving power that uses the wavelength of light. A wavelength is the distance between neighboring wave crests. A light microscope can only magnify and focus on an object up to a certain point. As the wavelength becomes smaller, the resolving power increases. Therefore, to see more detail, an electron microscope is used. We have covered quite a bit of information so far, students. For our next activity, let us review what we have discussed. Please define the following terms: magnification, resolution, and resolving power.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. I am sure everyone was able to remember all the terms. Let us review them together. The term magnification means to increase the size of an image of an object. The term resolution means the ability to distinguish between two separate points. The term resolving power means the amount of detail a microscope is able to show. Were you able to correctly define these terms? If so, great job. Let us compare the resolving power of the light microscope, the electron microscope, and the human eye, so we can fully understand the capability of the microscopes. The human eye can resolve to approximately 0.1 millimeter, if the objects are closer than this, the human eye will only see one object. 1 millimeter is equal to 1 multiplied by 10 to the power of -2 meters. A light microscope has a resolution limit of 200 nanometers. A nanometer is equal to 1 multiplied by 10 to the power of -9 meters. An electron microscope has a resolution limit of 0.3 nanometers. This is approximately 1,000 times greater than a light microscope. We have time for one last activity, students. Determine whether the following statements are true or false.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. Let us review the statements together. Light microscopes do not show the same amount of detail as electron microscopes. Electron microscopes show more detail than light microscopes. The greater the resolving power of a microscope, the more detail that can be observed. As wavelengths in a light microscope become smaller, the resolving power does not decrease. As the wavelengths become smaller, the resolving power increases. The smaller the distance that can be seen between two objects, the better the magnification. Were you able to answer them correctly? If so, great work. We have reached the end of our lesson. Today, we learned about magnification and resolution. We also compared the resolving power in light microscopes and electron microscopes. I hope everyone enjoyed the lesson today. Until next time, thank you, teacher. Thank you, students.